right. Let's get custom live streaming service going. All right. Okay, we're going to take a moment as people are logging in. Thank you so much. This is Popping the Science Bubble, hosted by Berkeley Public Library and in partnership with Cal. And we are delighted to host this program every month and get to meet new grad and postdoc and PhD students and hear them talk all about their research. Okay, so we're just going to take a moment to log in. And let's see, it looks like we are live on Facebook right now, too.
Yeah, so I can see us over there.
Hopefully. Are we live?
We are.
We are. Oh, good. Okay. All right. And I'm going to exit out of that tab and care of it. Right. Great. Actually, Madison, would you like me to watch the questions on Facebook this evening? I can do that.
Okay. Yeah, sure. Thank you.
Yeah, you bet. All right. So yeah, folks are just logging in right now. Let's see. Okay. It's been a busy day at the library. Lots of folks coming into our building for air conditioning. That is a service that we do offer at the central branch of the library. So if you're looking for a place to keep cool, I recommend it. We are starting to do more and more programs in person at the library, but we do not have evening hours yet. So we are not quite making the jump for Science Bubble quite yet. But we are hoping that one day this program will be in person once again. But I do encourage you to go to the library's website, check out what we've got going on. We've been hosting a series of pollinator events, talking about celebrating flowers and the insects and birds that pollinate them. I think the soonest event coming up for that is going to be on July 1st. And we are going to have Doug Talmy, who is a plant scientist, come in. I think he might be like leading a walk. Oh no, he's doing a virtual talk. Let's see. And he is an entomologist, conservationist, and professor in the Department of Entomology and Wildlife Ecology in the University of Delaware. And he is basically going to be talking about the decline in biodiversity and what people can do to help contribute to biodiversity and make our natural world a better place for bugs and bees and flowering plants. All right, so I think I'm going to go ahead and get started with our introduction here. Hello, everyone. Welcome to Popping the Science Bubble. This is a monthly seminar series that aims to share new research findings from grad students and postdocs at UC Berkeley with the general public and create constructive discussion about a variety of science topics. We have 2 speakers at each seminar who will talk about their current research or a topic that they find really interesting. The organizers are 3 graduate students at UC Berkeley, Jenna, Madison, and Oksana. Madison is with us here tonight. If you're interested in checking out past seminars or watching this seminar again, a video will be uploaded on YouTube. We are also streaming tonight's program live on Facebook tonight, and it will be living on our Facebook page. I just want to highlight again, this is a monthly program. It happens every 3rd Tuesday of the month at 5:30 p.m. And so we're just so happy for this partnership. Madison, I will let you introduce tonight's speakers.
Yeah, thank you, Kelsey. Thank you for everyone showing up. And of course, thank you for Megan and Jose being here to speak. We're excited to hear from you tonight. As Kelsey said, we are a monthly seminar series here through the Berkeley Public Library of researchers on the UC Berkeley campus. So feel free to stay tuned in month to month. We will also have an email list that you can get subscribed to if you find yourself interested tonight and want to hear future seminars. But for the format of the talk, I do want to point out that we are open questions. So please feel free as you're listening, if you have any questions about the research that our speakers are talking about, or any clarifications, anything at all, please feel free to add your question in either the Q&A or the chat box or the comment section on Facebook, and we will be monitoring and asking those questions to both speakers either in real time or toward the end of the talk. So please feel free to join in if you are at all curious. But with that, I'll introduce our first speaker, Megan. Megan was born and raised in South Florida and was a first-gen college student at the University of Florida, where she double majored in chemistry and Chinese. When she was there, she studied medicinal chemistry, which got her interested in chemical biology, and then did a brief summer research program at Penn State, where she was introduced to research understanding the roles of metals in biology. Here she was inspired to continue her graduate studies in bioinorganic chemistry, where she is currently a rising fourth-year chemistry graduate at UC Berkeley, studying a new family of metal-containing proteins in our cells. Outside of the lab, she enjoys studying foreign languages, playing the piano, and ice skating, and we're super excited to hear about her field. So please take it away, Megan.
All right, let me share my screen. Let's see. Okay. Thank you for the introduction and letting me speak here today. I'm really excited to tell you guys about metals in biology, which I find really interesting because so many important processes in life revolve around metals. And so understanding them and like what the metals are doing really just helps us understand more about what's happening in life. So I'll start with an example. We know that iron is in the blood and we need iron for life, but why? What is the iron actually doing? So for me to answer that, I first have to tell you what a protein is. DNA you can think of as a cookbook with instructions for a bunch of different recipes, and that can be transcribed to to a recipe card, and this is like the mRNA. And sections of the mRNA will code a little link on a protein, and you can continue to build the link until you have this protein chain made up of all these little beads. And then these beads will, I'll write them or draw it just as a line here for simplicity. They'll fold together, and this is now your protein, much like a finished meal. So our potatoes here. But sometimes we don't just want potatoes for dinner. So let's say we build another link and put it together. So these are two separate protein links, but stuck together to make a full protein. Much like you might have potatoes and steak to make your full dinner. And proteins are very important for a variety of reasons. They have a lot of functions. First, immunity, antibodies like the SARS-CoV-2 antibody shown here is a protein. And so this is just a more realistic depiction of protein folding. Sometimes if they're folded in this helical shape like this, this is keratin, which is important for structure and support in hair. And then we also have another depiction here of proteins, which are used for transporting different things across barriers in cells. And finally, one function that I find the most interesting is speeding up chemical reactions. So we have here, again, a more realistic depiction of protein folding, but often these proteins have little crevices inside of them where things can bind. Another cartoon right below that shows here something binding in the crevice. And inside of the protein, a chemical transformation can occur, and then it can be released out. And in the absence of the protein, that transformation will not happen. So let's get back to the iron then. What is the iron doing in the blood? Why is it important? So iron is in a protein that we probably have heard the name of, hemoglobin. This has 4 chains stuck together to make the hemoglobin, and each one of those chains contains an iron. And so the iron is actually sitting below a plane, and when it binds the oxygen, the iron gets smaller and is able to flatten, and flattening that plane tweaks the protein chain around, which causes the next protein chain to tweak, and it is able to bind oxygen even easier. Then the next one tweaks, can bind oxygen even easier, and the next one even easier. So this is what makes it so easy to transport oxygen, and then the release is the same kind of cooperative mechanism. So I think this is really interesting because it's a small detail of this metal that allows us to understand how oxygen is transporting around our body. But metalloenzymes can do, or metalloproteins, proteins containing metals, can do more interesting things than just transport oxygen around our body. So they're able to do these really interesting transformations, and nature has designed them in very exquisite ways. So they're able to do them, and it's really quite complex. So I'll give you a couple examples. The first is DNA synthesis. There is an iron-containing protein. I just showed here a picture of the protein folded, and there's iron buried inside of it. And it does a chemical transformation to make the building blocks for DNA. And this protein is found in every organism and every domain of life, you, bacteria, plants, everything. And it generates at the iron site a very reactive species, so reactive that scientists thought for years it was impossible for a living organism to have these kind of species because it would just kill everything. But the protein is able to control that reactive species in a really brilliant way. Another example is in photosynthesis. Photosystem 2 is the protein I showed here, this protein fold, and it's able to take water and convert it to oxygen and energy, which is the energy used in photosynthesis. And that conversion happens at a metal site. In this case, it's manganese. And almost all the oxygen we have here on Earth is produced through this, at this metal center, yet the mechanism is not fully understood. And then finally, natural products. These are sort of more complicated molecules, but medicinal chemists are really interested in being able to synthesize them because they can be useful for pharmaceuticals or different therapeutic applications. And metalloproteins, proteins containing metals, are able to do certain transformations in the process of making these natural products. And it's really quite interesting where you have all of these different any one of these different points is where the transformation could take place. But these proteins know exactly that it needs to happen in one specific spot every single time. And so the really complex nature of these proteins are able to do exquisite chemistry. So understanding them is interesting just from a fundamental knowledge perspective, but also can be quite useful to study metalloproteins. First, understanding something like photosystem 2 that I just discussed can help us learn how to devise more industrial applications. So how to use solar energy to create hydrogen fuel from water, which is a renewable energy source. Metalloproteins also serve as direct therapeutic targets because these proteins are in so many important processes in life. When these proteins don't function properly, they cause many diseases, including cancers, diabetes, and ALS, or Lou Gehrig's disease. If you don't know what that is, just think of that ice bucket challenge we were all doing like 10 years ago. And then also studying how these enzymes are able to do the chemistry, or the proteins, how they're able to do the chemistry they're able to do, can allow synthetic chemists to design more efficient routes to synthesize natural products in industry. So faster, cleaner, cheaper ways of doing these chemistries, these chemical processes ourselves.
Hey, Megan.
Yeah.
We do have a question in the Q&A. Sarah is asking, do metals play a role in neurotransmission?
I'm not very familiar with neurotransmission myself. I would definitely guess so, but I'm really not sure. I don't know about neurotransmission. Yeah.
Thank you.
Yeah. Okay. So I have one example to sort of drive home the practical application of studying metalloproteins, and that is this ribonucleotide reductase enzyme. This is the DNA synthesis enzyme I already mentioned that contains iron. And like I said, it makes the building blocks for DNA. So this protein functioning normally is important to have the right amount of each of the 4 DNA building blocks in our body. So you can think of these as pools of the different building blocks. But if you have too many of these, the cell can keep growing. You can make more DNA and you're going to have cell proliferation, and this is cancer. So But it's actually pretty interesting because scientists found that tumor cells are more susceptible. If you try to target this enzyme RNR, tumor cells are more susceptible to it because it needs that enzyme more to keep making more of these building blocks. And so there are already clinically active anti-tumor drugs that target RNR and others in trials. So that is one of these useful applications. And this brings me to a little scientist highlight. Joanne Stubby, shown here with me about 5 years ago. She was a professor, she's now retired, at the Massachusetts Institute of Technology, and actually got her PhD right here at Berkeley. And she was a pioneer in a lot of this RNR research. Her and her team developed a lot of the drugs that are in clinical trials now. And she was around at a time where science was mostly dominated by men. So she was really a pioneer for women in science. And she's also probably the most passionate scientist you'll ever meet. So she's really cool. Yeah. So now I want to talk briefly about what I'm actually doing in bioinorganic chemistry, studying metals in biology. So I show here these beads connecting to make a protein chain. And actually with modern technology, it's really easy to look at proteins and know what the sequence is or what the beads are that make up that chain. In fact, you see here in this chart, as of September of 2019, there's about 180 million entries where we just know the sequence of the protein, but that doesn't tell us how the protein folds, and it doesn't tell us what the protein does. And the only way to figure that out is by biochemically characterizing it, studying it in a laboratory. And proteins that have actually been studied in a laboratory are this yellow plot down at the bottom. So less than 0.3% of protein sequences have actually been studied biochemically. And so what we were thinking in our lab when we started my project, if we learned as much as we've learned studying the 0.3% of sequences we know already, what else can we learn about chemistry if we study any of the other proteins out there? And so we used some bioinformatics, and we found a family of similar proteins, about 15,000 proteins that had a very unusual fold compared to other proteins predicted to contain 2 irons. So these proteins in the 15,000 protein family was predicted to have 2 irons in them. And having an unusual protein fold, or this unusual structure, made us think it probably has different function, and we can learn a lot of chemistry from it because structure informs function. So if it's a weird structure, it's probably going to function differently. And so we wanted to look at these enzymes or these proteins and look at the structure. So how do you look at the structure of a protein? And I'll explain that using this egg analogy. If a protein is folded properly, it'll float around in solution, just like the clear part of this egg yolk. So egg is protein, and it's folded properly and it's clear. But when you heat it, actually what happens is the folded protein is going to precipitate and aggregate together. So it unfolds and it just aggregates, and that's why your eggs turn white. So that's aggregated protein. And so we do the same thing, actually. I have a tube of my protein, that's what I show here, and it's in solution, folded nicely. But instead of just cooking it, I do a very controlled, slow precipitation. So instead of breakfast, I get protein crystals. And my protein crystals, I can shoot them with X-rays. And the way that the X-rays bounce off of these protein crystals can tell us what the structure of the protein looks like. And that's what I'm showing here. This is the result of what those crystals told us. So basically, it tells us what are these beads and how do the beads connect. And indeed, like we thought, there is a very unusual protein fold for a diiron enzyme. So these like helices we're seeing, this is just the protein fold. And it's unusual in that it is really easy to evolve, meaning I could swap one bead for a different bead and give the protein a whole new function, or maybe enhance the function. And so it could be a really convenient platform for engineering in the future. Also, interestingly, the beads that were holding those irons into place were pretty unusual. And like I said before, if we have unusual structure, so unusual types of beads holding it there, we're going to have unusual function. And different function is what we want to learn new principles of chemistry so we can understand how these reactions take place. And through my research, I found that these transformations that these enzymes are able to do were actually very difficult. So understanding how they're able to do it is very useful. And finally, actually, these enzymes don't have 2 irons like was predicted from the original bioinformatic analyses. They must have 2 different metals in the active site, which is completely unprecedented for these types of proteins. So that is a whole nother avenue that we're looking to explore. So what we're doing right now, we are understanding how the enzyme or how the chemistry works, comparing the structure to the function. And hopefully we can eventually extrapolate these chemical principles that we learn for either industrial applications or protein engineering. And so with that, I just want to briefly thank or acknowledge my lab, my mentor, Jonathan Riddle, my lab mates, our collaborators, and NSF for funding, and you guys for listening. And yeah, I'll be happy to take any questions.
Great. Thank you so much, Megan. As any of our audience members want to put questions either in the chat or the Q&A, I might start us off. So given that normally if things have very similar structure, they do very similar functions, are you finding that these like over 15,000 proteins are all very similar? Does that mean, like, I'm assuming they're across organisms rather than all within one?
Yeah, that's a great question. So they are across different organisms. And so our folds actually have, there's a lot of similar parts, but there's these weird loops in between that are quite different across the different types of organisms. So they're doing similar chemistry, but the type of substrates they're working on is very different across. So we have natural products, that chemical transformations of natural products. There's small amino acids, large sugars. So there's actually quite a lot of diversity.
And then maybe a follow-up to that, are any of these like chemical processes that we didn't know about making like an organic molecule, like a sugar that we just don't really know how to make super well? Are there any like fun ones that you found that have been like, we never knew how we could synthesize that? I'm excited about being able to potentially synthesize that.
Yeah.
Sorry, that's a big question, I know.
So one of my lab mates, she's studying one enzyme, and that's doing a chemical transformation in a biosynthetic pathway that we don't know how to do that reaction with just like synthetic catalysts. So there are definitely examples of that in this family. The enzyme I'm looking at or focusing on, I wouldn't know how to make it synthetically either, actually, but it's not too complicated. But it's the speed and the, I guess, precision that the enzyme can make it is sort of what makes that a pretty interesting reaction to study.
Cool, thank you.
Yeah.
We have a question coming in through the Q&A asking, how did you learn to visualize protein geometry when their structure appears so complex?
Yeah, that was sort of in the process of solving it. So it's complex when we zoom out, but there are a lot of softwares where you zoom in and see all of these, what I keep calling these beads, they're actually amino acids. And so we zoom in and we can see all of the atoms, and just looking at it a lot. Yeah, it becomes a little bit easier. Obviously, when you're looking at it zoomed out, it kind of means nothing, even to somebody who's looked at it for a long time. Until, yeah, zoom in and look closer at each atom.
Cool, thank you. And did you know crystallography or how to determine structures from crystals before you got to grad school, or is this a new venture? Yeah, crystallography is completely new since I've been at Berkeley. Yeah, it's pretty fun though. Yeah, congrats. That's a whole big thing. Getting crystals even is very exciting.
Yeah.
All right. If there are no more questions coming in through the Q&A and chat, I think Megan will still be around through the second talk, so you're free to ask any questions through the chat, and I think she can answer them as they go, and we'll also have a short little bit of time toward the end of this webinar for any further questions. But now I'll let Jenna give the introduction for our second speaker.
All right, So for Jose is a PhD student investigating age-related changes to skeletal muscle mitochondria. Jose was born in Guatemala and moved to Los Angeles when he was 3 years old and grew up in the Boyle Heights neighborhood of East Los Angeles. Outside of the lab, Jose likes to play with his dog, Cena, and go on hikes and trail runs and play rugby. So Jose, take it away.
Thanks, y'all, for the opportunity. Let me quickly share my slides here. Okay. Does that look okay?
Yes.
All right, cool. So welcome. Hello, my name is José Arevalo, PhD student in the Department of Integrative Biology. And yeah, I am studying the aging mitochondria and just really looking at how this powerhouse of our cells are lost as we age. So to start, what is the mitochondria? Well, I mean, if you just look at your biology books or just any search up mitochondria, you'll see the mitochondria as these little sort of oval-shaped organelles. And so they are these oval-shaped organelles, or in this case, it's one big organelle, but I'll get into that later. That is part of your each one of your cells. And what does it actually do? So if you just look at the actual structure, you zoom in, and it actually has two different levels. So it has an outer level, which is the outer membrane, and the inner level, and it really is the inner level that contains these specific structures in which you have what's called an electron transport. So it's just movement of electrons in order to create energy. And how does it do it? Well, it metabolizes different molecules. So for instance, you have a metabolism of a sugar, right? So I just have a donut here. You have a sugar being broken down, and the very end product of this sugar is used to allow these electrons to transfer through these structures of the inner membrane of the mitochondria. You also have fat. You can either metabolize sugar or fat. And again, the breakdown of these structures sort of travel through the inner membrane to allow for the mitochondria, for the electrons to pass through these specific structures. And so that's essentially what the electron transport chain is that it allows this movement of electrons either through breakdown of sugars or breakdown of fats to consume oxygen and make what we know is what we call ATP, but I'm just going to call it here as energy, right? And what is energy actually used for that the mitochondria makes? Well, in order to do that, I'll just quickly talk about the skeletal muscle. And really, this energy is used to provide energy for the skeletal muscle, right? And so skeletal muscle moves our bones. And so we have skeletal muscle here is sort of a collection of these long cylindrical cells that are made up of specific proteins. And each protein has a section called a sarcomere. And these specific sections actually come together for the contraction of your muscle to be able to move these cells. So I know there's a lot going on here, but I just want you to focus on the relaxed muscle is sort of a extended muscle. And so you have these proteins sort of away from each other. And the contracted muscle is the shortened muscle. And so you have the proteins overlapping and closer to each other. And you can see that ATP, this energy, is what is being used in order for muscle to actually contract. So it's called the powerhouse of the cell because it actually powers your skeletal muscle cells so that you're able to move, right? So this specific energy actually moves you. So how do the mitochondria actually stay healthy? Well, they go through what's called the mitochondrial dynamics, which is just continuing restructuring, this breakdown or this division called fission, and this continual elongation called fusion. So it's sort of this restructuring of the mitochondria, which are controlled by various proteins. And fission, this breakdown, actually does occur with stress and with damage to these layers of the mitochondria. So this stress is actually caused by what are called radicals. And so if we think of sort of these proteins or these structures that move across the inner mitochondrial membrane in order for oxygen to be consumed so that it makes our energy, we have these electrons that actually bind to oxygen before oxygen actually arrives to the specific structure called the mitochondrial complex 4, which is, if you can see here, it's where oxygen is being consumed. And so these radicals are actually generated there before they get to that last structure where oxygen is being consumed. So complex 4 is where oxygen is consumed, but before oxygen is consumed in that complex, these certain electrons actually bind to oxygen to make these specific radicals and cause damage to the mitochondria. So how do they actually stay healthy, especially if they do get damaged? Well, it's a term that's called mitophagy, and it's a destruction or eating of the mitochondria. And so again, as I mentioned, you have this continual restructuring, the fusion and the fission. And in order to make sure that the damaged mitochondria doesn't continue to cause any more damage to the cell, you have sort of this mitophagy occurring so that you maintain this health of that mitochondria. So as we get older, more of this fission, more of this division actually occurs. And because of that, we also have this reduced ability for mitophagy, for this removal of this damaged mitochondria. So we have sort of this cycle of more fission, reduced removal of the damaged mitochondria leads to more fission and reduced removal of this damaged mitochondria. Sort of this continual cycle of just bad mitochondria leading to more damage, leading to less activity of this mitochondria. In addition, talking about skeletal muscle, as we get older, skeletal muscle actually decreases, meaning the muscle mass decreases of our skeletal muscle. And so if we think of that age-associated muscle loss, the term for it is called sarcopenia. So if we think that energy from the mitochondria allows our muscles to move us, if we have less of this muscle, then we will have less ability to move our bones and move our cells. So less muscle means less energy, means less movement. And because we're talking about this energy being ATP, that means that because we have less muscle, we probably also have more damaged mitochondria. We also have less production of this energy, production of this ATP. So one of the studies here that I'm just showing is that when you have a comparison, there's a lot going on here, but I just want you to focus on the mitochondrial synthesis of this energy. So when you compare young participants, young healthy participants, the old participants generate less energy compared to these young participants. And as we get older, we also consume less oxygen. And so this is a whole body oxygen consumption. So in order to measure oxygen consumption by our muscle, we actually sort of take measurements via our breath. And so this is a picture here of myself getting my breath measured with increasing exercise. And so, yeah, as we get older and we test different aged participants, we should be able to see a reduction in this whole body oxygen consumption.
Jose?
Yeah.
Can I ask, when we get older, are we worse at like taking in oxygen or just worse at efficiently using it?
Yeah, and in a sense, both, right? So we also do have a reduction in our sort of lung function. So if our lungs are not functioning correctly as we get older, meaning that we also have a skeletal muscle that holds our lungs, expands our lungs so that we can take in oxygen. So that muscle is also, if we have loss of that specific muscle that expands our lungs, which we do, then that also reduces our ability to inhale and take in oxygen, not just the use of that oxygen.
Cool, thank you.
Yeah. So where do I come in? So my focus really, and my question overall for my PhD is, how do the changes to mitochondria relate to this reduction in oxygen consumption, right? So I showed you that there is changes to mitochondria as we age, and with those changes, there is a reduction in oxygen consumption. So where is that specific relationship? And what I want to do in order to figure that out or answer that question is I want to use specific skeletal muscle progenitor cells, and these are called precursor cells that are developed from skeletal muscle stem cells. So if you think of what stem cells are, these ones are specifically programmed to become skeletal muscle. So that's this image I have here. So this is an image I took of one of these skeletal muscle progenitor cells from a young individual. And I stained for the mitochondria. So this is a mitochondrial network here in red. And I also stained for the nucleus. Whoopsie. Here we go. And I also stained for the nucleus, and it's the nucleus here in blue. So it's just one of the images that I want to see. What are the changes to the mitochondria, and how does this related to oxygen consumption using these specific skeletal muscle progenitor cells? So if you think of these progenitor cells, these are cells that come from, as I mentioned, skeletal muscle stem cells, also called satellite cells. And these are actually important for recovery after injury. So when you have an injury, you have these specific satellite cells that actually come in to try to make more of these specific skeletal muscle progenitor cells, and the skeletal muscle progenitor cells fuse with the fuse and aid in recovery of this injury and damaged skeletal muscle. So what I really want to work with is this second stage here. So you have the very first stage on the left, which is the satellite cell, the muscle precursor cell, or the stem cell. And then those are developed into what are called myoblasts. And these are the specific progenitor cells that I'm using for my mitochondrial studies, right? So looking at these for changes to the mitochondria and looking at these for changes in oxygen consumption. What I have so far as looking at these changes in these specific myoblasts or these progenitor cells is that myoblasts from old individuals have lower oxygen consumption. So this is a test where I measure oxygen consumption based on the stress that I apply to the mitochondria. So I just apply some stress and it gives me these different readings. And so the blue line, as you can see here, are these progenitor cells from a young. The red line are the progenitor cells from an old individual. And you can just see that there is a lower oxygen reading, or the line is much lower compared to the line from progenitor cells of the young. In addition, using this same data, I can calculate ATP production, and so lower production of energy, of the specific energy, also occurs in these myoblasts or these skeletal muscle progenitor cells. So what can you do, right? So I did mention that, yes, mitochondria are, mitochondrial function is going to decrease, oxygen consumption is going to decrease, your energy production is going to decrease. What can you actually do to help you relieve or prevent this age-related changes? Well, for one, really be active, right? There was a study that looked at active participants and non-active participants between the years 60 and 80 years old, and older individuals that were more active had a higher mitochondrial volume density. So if you see this top figure here, it's MitoVD, mitochondrial volume density, and also more mitochondrial, or excuse me, much better mitochondrial functioning. So these are just the different complexes of the structures that I showed you of the electron transport chain, and there's more of these structures in active old individuals compared to sedentary or non-active old individuals. So exercise actually does stimulate biogenesis. It stimulates this fusion process, and it also stimulates mitophagy, which again is just removal of these damaged mitochondria. And this is just me being a little, what's the word I'm thinking of here? Biased as far as the type of exercise. Some focus on resistance training, right? So as I mentioned, skeletal muscle you do lose some skeletal muscle as we age. And so resistance training actually does help prevent that skeletal muscle loss with age. You don't have to go to the extremes with the powerlifting grandma to deadlift 255 pounds at such an old age. But there are classes that I found at the downtown YMCA that do that have classes for senior strength activities, so that you can at least focus on, again, on resistance training and help as we get older to prevent this skeletal muscle loss. So thank you to Dr. George Brooks and Dr. José Vasquez Medina, and of course, all the members from the laboratory of Dr. Brooks and Dr. Vasquez Medina. And thank you all for listening. My email is on the screen currently, and this presentation is now open for discussion.
Awesome. Oh, yeah. Thanks, Jose.
Yeah, thank you. Sorry, I think Jen and I both went to moderate questions there. Yeah, we're happy to take any follow-up questions in the chat and Q&A. I was wondering, so when these mitochondria break off, right, they normally, or fission, they normally break off kind of the damaged bits and sequester them. So when you can no longer recycle them, you end up just kind of like accumulating. I guess basically you're sitting around where like your waste bin is much more full more of the time. But does that actually also result in the rate of, as you get older, does the rate of your mitochondria getting sick also increase? If that question makes sense.
Yeah, so that was that one slide about increase in that fission, right? So it's the increase in more of this rate. So there's less ability for mitophagy, which is removal of the damaged mitochondria. So then you just have this increase of sort of this continual division. And so that, you know, that along with more damage sort of leads to this lack of function of the mitochondria.
Okay, so the seesaw's tilted on both sides, kind of.
Yeah.
All right. We have a question in the chat asking, are the muscular precursor cells generated in the bone marrow? No. So these are essentially, you know, you just have this precursor cells that are these stem cells that are specific to mitochondria. And so, I guess I don't remember at the moment here, because if you're thinking of just stem cells, but yeah, I'm gonna say if they're programmed stem cells, programmed to be skeletal muscle cells, then yeah, they would be essentially, they would have that same, they would come from all of those stem cells and then just become programmed to be skeletal muscle cells. But I apologize for not being too sure at the moment. Great, thank you. And then we have another question asking, I noticed you didn't mention nutrients that are allegedly antioxidants. Are you dubious of that?
I did not, no. And that was just because I thought I was going to take long with this presentation. But yeah, there definitely are ways to also mitigate the radicals, or at least be able to flush them and prevent them once they are generated. And those are different antioxidants. And so, yeah, that was a very good question. I thought I wasn't going to have time to present those, but yes, there are antioxidants out there. And nutrition is pretty important as well to mitigate these effects of mitochondrial damage.
Cool, thank you. I was wondering if you see in skeletal muscle progenitor cells, if they also have a really high rate of making new mitochondria, right? Like if you recycle, not as you get older, just in general, does this cell type have like a large demand for, as you turn over damaged mitochondria, making new mitochondria?
Yeah, so these are, so being that they're precursor cells, they are, they do grow a little more fast than you would say normal cells, which means that they do require more energy consumption, which means that there is sort of a more of a mitochondrial, what's the word I'm thinking of here? Yeah, I apologize. I can't think of the word that I'm thinking about.
Biogenesis, I think.
Yeah, there we go. Yes. Yeah, so that's one thing that I'm hoping to also look at is, you know, like how different, if there is changes to mitochondrial density and more fission as we get older, do I also see this in these progenitor cells from from progenitor cells that were taken from old? I mean, I do see changes in oxygen consumption, but at the moment I don't see any of those changes to the mitochondria network itself.
Okay, great. Thank you, Jose. If there are any last minute questions, everyone can feel free to type them really quick in the chat and Q&A. But otherwise, I think both of our speakers have made their contact info available if you have any follow-up questions, and we will of course send a post-seminar email with the resources our speakers have greatly or generously shared with us, as well as post these talks to YouTube in case you are interested in rewatching or sharing with friends, family, or anyone else you know that might be interested. So thank you very much, Megan and Jose, for speaking with us tonight. We really enjoyed hearing both of your talks, and we appreciate you guys coming out. And I'll give it to Kelsey to wrap up.
Yeah, these were excellent presentations tonight, and I thought they complemented each other nicely too. So thank you so much. Just a reminder, these talks will be going on YouTube, and Science Bubble happens every third Tuesday of the month at 5:30. Thank you, Jose. Thank you, Megan. These were just great presentations. And I am going to go ahead and sign off. Have a good evening, folks.